Crawford, and today I'm going to discuss my research paper titled Differential Responding Using Multiple Schedules of Reinforcement. Multiple schedules of reinforcement are schedules that are alternated based on time. Unique stimuli are then assigned to each schedule. Multiple schedules in general have been further explored in a greater variety of socially significant applications, such as the classroom setting. Brief signals prior to the schedule have shown to be effective in implementing multiple schedules to promote differential responding. The present study seeks to build on previous research of multiple schedules of reinforcement and its impact on differential responding. For the present study, a male North American betta fish was used for the study, approximately 1.5 inches in length and purchased from a local pet supply store in the researcher's hometown. For the study, the previously described North American betta fish was used in the study. Upon the fish's arrival to the researcher's home, a 2.5-gallon tank was prepared in order for the fish to have a safe and livable habitat. The water was treated with 100 small drops of approved water conditioning solution and allowed to sit and maintain room temperature for 24 hours prior to the fish's entrance. The tank was also purchased from the local pet supply store. A small artificial plant was placed in the back right corner of the tank. The R2 fish school kit was purchased and acquired by the researcher in order to assist with the training process. The researcher used the provided long feeding rod in order to provide the fish with food as reinforcement for the procedure. In order to signal the two different schedules of reinforcement, two built-in features of the researcher's iPhone 8 were used. The first feature used as a signal was the vibrate feature. The vibrate was held up against the tank and lasted approximately half a second during each corresponding trial. The second feature used was the flashlight feature. This flashlight was used for 1 second during each corresponding trial. The behaviors being trained in the study were swimming through a hoop and swimming through a tunnel, both of equal response effort. The North American betta fish was taught to receive food reinforcement from the aforementioned feeding rod. During the training, the fish was fed exclusively during training times and only via the feeding wand. The fish was kept in a closed economy throughout the duration of both baseline and intervention phases. The fish did not receive any extra food outside of the training sessions. Once the fish reliably demonstrated the ability to swim up to the wand in order to receive food over 5 consecutive trials, the two apparatus, the hoop and the tube, were introduced to the fish. The fish was taught to swim through one of the hoops, initially with maximum prompting from the wand. The feeding wand was then progressively faded as the fish continued to demonstrate responding of swimming through one of the two hoops. Once the fish demonstrated independent and consistent responding of swimming through one of the two hoops, baseline data was collected. During the baseline phase of the experiment, the fish was reinforced for swimming through one of the two hoops on an FR1 schedule. The fish received one small pellet of food for each time the behavior was demonstrated. Baseline was conducted for a total of 3 sessions over 3 consecutive days. Baseline responding did not show any differentiated responding in the fish, as the fish chose to swim through one of the two hoops at approximately the same rate. Once baseline responding was deemed to be stable, each of the two behaviors were assigned a correlated external stimuli to be produced by the researcher. Swimming through the hoop was assigned the external stimuli of a short vibrating sound. The vibrating was assigned an FR3 schedule. The fish was reinforced for every 3 responses swimming through the hoop. Swimming through the tunnel was assigned an external stimuli of a quick flash of light from the researcher's cell phone, placed closely to the wall of the tank. The flash of light was assigned a VR3 schedule. After each trial in a given session, the signaling stimuli were alternated. For each first trial in the session, the opposite signal was used to limit any potential bias in the schedules. During each session, each signaling stimuli was used a total of 5 times as data was collected from the lone researcher. Here we have a video of the fish's pre-training responding with both hoops being in the tank and the fish not responding to either one of them. This video is taken prior to baseline data being collected. During training, the fish needed maximum prompting from the feeding wand in order to engage in the target behaviors. The wand was held about halfway through both of the hoops in order to help promote independent responding. The wand was then faded as the fish demonstrated more reliable responding. Once training was completed, the fish associated the stimuli with swimming through the hoop and did not need maximum prompting. Oops. As mentioned previously, baseline data did not show any differential responding as the fish was reinforced for each behavior on a continuous schedule. The fish engaged in each of the two behaviors in equal amount. Over 10 total baseline trials, the fish chose to swim through the hoop 5 times and the tunnel 5 times. A multiple schedule of reinforcement was then implemented where 2 schedules were alternated, each being correlated with its own stimulus. The schedules and correlated stimuli were alternated over the course of 10 trials over 5 separate sessions until differential responding was shown by the fish, as demonstrated by the increased response of swimming through the hoop on the left when compared to the swimming through the tunnel on the right. Over the course of the 10 trials, the fish demonstrated differential responding by engaging in a swimming through the tunnel at a higher rate than swimming through the hoop. The fish swam through the tunnel at almost twice the rate of swimming through the hoop. This research builds on previous research that brief signals can be an effective stimuli in communicating the varying multiple schedules. This study demonstrated the effectiveness of multiple schedules to produce differential responding in an organism. Although the study was done in a closed economy, the results were consistent with previous research. Further research should be completed concerning the use of signaling stimuli and multiple schedules.